This last section in the asexual cell division segment of your notes is on cell cycle checkpoints. And before we go any further, I'd like for you to draw, hopefully from memory, the cell cycle with all of the different segments that we have talked about. I would like for you, when you draw mitosis, to actually draw in the stages of mitosis, really just PMAT. So you're drawing interphase, you're drawing mitosis, you're drawing cytokinesis in that cyclical diagram, so you could refer to the diagram in your notes. I don't want to go on until you've drawn it. So if you need to pause the video, go ahead and pause it. The goal is for you to see if you remember the parts of the cell cycle before we start talking about checkpoints. Checkpoints are going to be regulatory stages that must be passed before proceeding into the next stage. So we're actually just going to draw this. Instead of taking any notes or printing anything off, you're really just going to draw this on your own, because the diagramming itself should be pretty important for you to remember. So if we were to take our circle that represents the cell cycle, not bad. We keep in mind that interphase takes up the majority of this time in the cell cycle. So we would have just a very small segment of the cell cycle that would not be considered interphase. And one of the reasons for this is if we are breaking down the nucleus, moving around the DNA, we want that process to actually happen as efficiently and quickly as possible, so there's less time for mutations or damage to the DNA. So what we should have is the concept that all of this is interphase. But that interphase is going to be divided into segments. So you might have drawn this any way you want. Hopefully you kind of identified that we would have a portion called the G1, or the gap 1. Then we'd have a portion called the S phase, and then we'd have a portion called the G2 phase. And so when we look at the cell cycle, we know that this is where the cell is going to be kind of living its everyday life. And if it gets some sort of signal to divide, it will next synthesize its DNA. Well, when we get this signal, this is actually where we'll have our very first checkpoint. And it's called the G1S checkpoint. The G1S checkpoint. And at this checkpoint, we are making sure that we have enough materials to synthesize the DNA. So we're going to look at the environment, both internally and externally. And we're going to see if we have enough materials to actually copy the cell. We're going to really look at, is the cell capable of doing this? Is the cell big enough? Does the cell have everything that it needs to go into the synthesis stage? So we've talked a lot about enzymes, and we've talked a lot about inhibiting enzymes. And so it turns out that the main regulation is going to come from what we call CDKs, cyclin dependent kinases. And if you go back to the picture that you drew on page 220, figure 11.17, it actually is a picture of a cyclin-dependent kinase. these kinases are going to phosphorylate, add a phosphate group, and we know that will change the shape, and in this case it will activate other molecules, usually other enzymes. So the big concept here is that the cell is going to be controlled by enzymes that activate other enzymes. And this kinase depends on something else called cyclin. And cyclin is going to be a molecule that increases in concentration during cell division. So as cell division proceeds, I see more and more of this cyclin. I activate more and more enzymes. There are tons of different CDKs. If you Google CDK, you'll come up with, lots and lots of different examples. Some of these are going to activate enzymes. Some are going to encourage the DNA to coil up. So lots of different uses for these CDKs. So we'll see them at all the different checkpoints. So going back, the first checkpoint is this G1S checkpoint. And we are going to see CDKs here. They are what is actually doing the regulation. If what we need is present, then they get activated and they activate other things and we proceed. If we pass the checkpoint, then we go into the synthesis stage. If we do not pass the checkpoint, there are three options about what can happen. We can wait. And this is going to be what we call G0. This means that the cell is not capable of dividing, but it's capable of living out its life. We can amend or fix the problem. So if I don't have enough nucleotides, maybe the organism does some more consuming or producing and ends up with more nucleotides. Or it might turn out that the cell is damaged and we can trigger apoptosis, which is cellular death. So each time we hit one of these checkpoints, we'll either pass the checkpoint and go on, or we won't pass the checkpoint. And if we don't, we might keep the cell functioning just as a workhorse. We might try to fix it so that we can replicate the cell. Or we might decide that the cell is too damaged and we might actually trigger cell suicide. Moving in, we go to synthesis. We go to gap 2 where we grow some more. And we hit another very important checkpoint. And this is the G2M checkpoint, because it's going to separate us from interphase and mitosis. What's going to happen here is we're going to again check the cell to make sure we have everything, especially that DNA was replicated correctly. And so we're going to have lots and lots of different enzymes here, proteins as well, that are going to check the DNA to make sure it was replicated. One really important one is called P53. It is supposed to actually check DNA to make sure that it was replicated correctly, and if not, sometimes it can fix the DNA. So the G2M is still checking the cell mostly internally. We're really focusing more on the inside of the cell. We've already decided that the environment externally is okay. We've copied the DNA, we've grown the cell a little bit more. We're really checking internally. If we pass, again, it'll be the CDKs that do most of the work. But we do have a complex called the MPF. The MPF, which is the maturation promoting factor. So it's gotten a little busy over here. My screen is only so big. So, and I can't write down here at the bottom, so maybe you write down here at the bottom. But we have what's called the MPF, which is the maturation promoting factor. It turns out that we can actually think of it as the mitosis promoting factor because we'll actually move into mitosis.
Mr. Sutton, please call the front office. Mr. Sutton, please call the front office.
And basically this is a collection of CDKs that are going to encourage us to move into mitosis. So it just so happens that we have a name for the collection of CDKs here, and that's the MPF, or maturation promoting factor. You can also think of it as mitosis. So once we have entered mitosis, we talked about how one of the big concepts is what happens in anaphase. And the theory that we really can't go back, we can't reverse separating the chromosomes very easily. So each of these checkpoints, you'll notice, is before a really big event. Before I copy the DNA, before I break down the nucleus. And so in mitosis, the big stage is actually going to be, we've got PMAT. The big checkpoint is going to be here, between
Aaron
metaphase.
Whitley, come to the front office, please. Aaron Whitley, please come to the front office.
Between metaphase and anaphase, and so we call this the metaphase checkpoint. What we're checking for here is spindle attachment. Are the spindles correctly attached to the kinetochores? If they're correctly attached to the kinetochores, then I can start to allow the doubled chromosome to be pulled apart, the sister chromosomes, chromatids, to be pulled apart. It turns out that they're actually proteins holding them together. And what we have here is what's called the APC. APC is the anaphase promoting complex. The APC is the anaphase promoting complex, and that will actually encourage us to move from metaphase into anaphase. After that, that's the rest of the checkpoints. After that, we will continue on. We will finish with telophase, and then we'll move into cytokinesis. So your three big checkpoints are your G1S checkpoint, G2M checkpoint, and your metaphase checkpoint. The majority of these are going to be regulated by cyclin-dependent kinases. If everything in the cell is meeting the expectations, then we'll start to phosphorylate or activate other molecules so that we can do things like copy the DNA, break down the nucleus, separate the sister chromatids. So the big thing here is thinking of what is required for me to pass each of these checkpoints. And why are the checkpoints where they are? And what happens if I don't pass the checkpoint? So what is the checkpoint? What has to happen at the checkpoint? Why do we have to pass that checkpoint? Why is that checkpoint there? And then what would happen if we didn't pass? Those are kind of the big